Our second grant recipient is Dr. Niclas Bengtsson, Assistant Professor in the Department of Neurology at the University of Washington School of Medicine in Seattle, for his project entitled Development of Gene Regulatory Cassettes that Enable Safe and Efficient in vivo dystrophin gene editing in muscle stem cells. Dr. Bengtsson, I'll pass it over to you.
Thank you, Dr. Hoffman. Well, good evening, everyone. Can you hear me?
Yep, all good. Thank you, Dr. Benson.
Excellent. So I would also like to thank the Justice Journey very much for funding this exciting project to develop gene regulatory cassettes to make it possible to use CRISPR-Cas9 to edit muscle stem cells. One of the main features of Duchenne here is this ongoing muscle injury and regeneration. And it's due to these unstable muscle fibers that because they don't express any dystrophin. And here you can see these individual myonuclei that make up these large myofibers or cells. And on top of each one here, we have multiple what's called muscle stem cells, satellite cells, or as I'm going to be abbreviating them later on, SCs. In Duchenne, because there is no dystrophin, you have these this instability that when there is force produced by the fiber, you get this injury that causes the fiber to degenerate. And you end up losing these myonuclei, and that activates these muscle stem cells that starts to divide and fuse into these fibers to then regenerate the fiber. Unfortunately, these regenerated fibers still do not express dystrophin, so you have this cycle of ongoing injury and regeneration. One way to deal with this is using gene therapy via the use of adeno-associated viral vectors. Now, these are excellent from the ability of being able to reach muscles through the circulation. They're also not known to cause any disease in humans, and they do not interfere with the patient's own DNA. Some of the limitations, though, is that they have a relatively limited packaging size of less than 5,000 base pairs, and the fact that any future repeat treatments could be tricky based on an immune response to a previous exposure to these AAV particles. So for Duchenne, the normal full-size dystrophin gene is too large to fit inside these AAV particles. But these highly functional smaller dystrophins called micro-dystrophins that have been developed by my mentor, Jeff Chamberlain, and others, can easily fit inside these AAV vectors, and their expression can be controlled in using these muscle-specific regulatory cassettes that has been developed by Stephen Hauschke here in our group, who is also a collaborator on this project. And that would enable these microdystrophins to be expressed exclusively in muscle and not in non-muscle tissue. Now, how this works is that these AAV particles would then be delivered and they will reach these muscle fibers where they would unpackage and go into an individual myonuclei, where these genomes would then start to produce dystrophin to then stabilize these muscle fibers. However, microdystrophins are not quite as effective as normal dystrophins. So either due to trauma or normal muscle maintenance, what you can have over time is this gradual replacement or repair of these myonuclei that could cause the loss of these AAV vector genomes as well as the microdystrophin expression, leading to this loss of stabilization of these myofibers. Now, CRISPR-Cas9 or gene editing is an attractive alternative where one can use the bacterial Cas system, which pairs with a guide RNA and can be targeted directly to the error in the dystrophin gene that causes DMD. And then we can repair the patient's own gene, resulting in a larger and hopefully improved functional and stable protein. This can also be packaged into AAV vectors, like we have shown on a couple of occasions, and be delivered systemically. However, at least in my book, this in vivo gene editing does require added emphasis on a precise editing control to limit any permanent side effects that might occur here. And this will be done by using muscle-specific regulatory cassettes, which also coincidentally is important for limiting immune responses to the Cas9. Similar to micro-dystrophin gene transfer, here we would deliver AAV vectors containing this CRISPR-Cas that would go in to these myofibers, and the AAV vector genomes would make CRISPR-Cas9 that would correct the patient's dystrophin gene, leading to the production of normal dystrophin. However, these repaired myonuclei could still be lost over time, perhaps slower than for microdystrophin, but especially if we are unable to target these muscle stem cells. Now, this is a unique feature of CRISPR-Cas9, being able to target these stem cells, but so far today, there is no specific, efficient, or safe methods to target these cells today. And therefore, that is the goal of this proposal, is to develop these muscle stem cell specific regulatory cassettes. Now, looking at muscle stem cells on these fibers, normally you have these resting stem cells, and they express a gene called PAX7. When you have injury to these muscle fibers, these stem cells respond by being activated and turning into these activated stem cells that now not only express PAX7, but they express another gene called MyoD. These cells then divide rapidly and become this large pool of stem cells that then go on to fuse and repair the myofibers. Interestingly, a small amount of these active satellite cells can then return back to the resting stem cell phase and start expressing PAX7 again and be ready for subsequent rounds of regeneration if called upon. So our approach here, our first one, is to look at this PAX7 gene and look at the regulation of it. And here we're going to be trying to isolate these regions that control the expression of PAX7 and assemble them into different regulatory cassettes and use them to drive Cas9. We'll package this into AAV vectors that then we will deliver to go into these resting stem cells, correct the mutation, and then we'll get these amplifying pool of active proliferating stem cells that then fuse to repair myonuclei that are able to express dystrophin. And like I just mentioned, a small part of these cells are then able to go back into this resting stem cell state and be ready for future rounds of regeneration. Some of the advantages of this approach is that it has a relatively large downstream effect as we are targeting pretty early on in this cascade. Another advantage is that it can be used without injury. So it could be applicable to other forms of muscular dystrophies as well that don't have quite the regenerative involvement that Duchenne does. Possible limitations is that PAX7 would always be on in these resting cells that end up keeping these AAV genomes. And the fact that PAX7 is not necessarily unique to muscle. It's also expressed in testes and in brain, though we think that this is due to different enhancers, which we aim to eliminate as we start looking at these regulatory regions. Our second approach is going after these active or proliferating stem cells by looking at regulatory regions involved in the expression of MyoD. And here we would use AAV to correct the dystrophin error in these active proliferating stem cells during a very narrow window in time. And then the cells that are corrected would then continue to divide, fuse to form myonuclei that can produce dystrophin. And even here, these can also go back to become resting stem cells with the dystrophin correction and be ready to contribute to future rounds of regeneration. The advantages of this approach is that MyoD is actually only expressed in muscle, so it would be a little bit more specific. Also, as I mentioned, this will, since we're targeting this transient amplifying pool of active stem cells, the production of Cas9 would only be temporary and would not be expressed in these myonuclei or in resting stem cells. A possible limitation here is that it could have a slightly less impact in the absence of injury, as we would require there be a large amount of these active proliferating stem cells in order for it to work. But that should work great for Duchenne. So just to finish up here, I wanted to give you guys a brief project overview. We will start here by designing an in vitro testing and figuring out these different regulatory cassettes and try them in myogenic stem cell cultures and compare them to control cell lines. Once we have a number of candidate regulatory cassettes, we'll move into this in vivo testing where we would use AAV particles to transduce muscle, and then we'll harvest these targeted stem cells, and we'll compare how well these regulatory cassettes work in those cells. We foresee this being an iterative process, going back and forth between the design and in vitro testing and in the in vivo optimization until we have established a number of prime candidates, at which time we will then move forward to our final phase, which is to cross-validate these regulatory cassettes in large animal models of Duchenne muscular dystrophy. So with that, I would just like to introduce some of the collaborations and the resources that are available to complete this project here at the University of Washington. And with that, I say thank you.